Hello, this is Dylan and James. Our cell communication project will be on hormone signaling. To start off, hormone is a class of chemical that is generally regarded as produced by glands and has important effects on organisms. They are generally transported via bloodstream from the cells that secretes them to target cells or a group of target cells. Hormone signaling includes the transportation of hormone via bloodstream, recognition and reception by the target cells, signal transduction within the target cell, as well as the final genomic and non-genomic effects on the target cells. Hormones includes three subclasses: amino acid derivatives, eicosanoid hormones, and steroid hormones. Amino acid derivatives includes peptide and proteins, of which amino acids are the building blocks. Eicosanoid hormones are oxidized fatty acids, for instance arachidonic acid and lipoxin. Steroid hormones are all derived from the 4-carbon ring lipid cholesterol. The main difference between steroid and non-steroid hormones is that steroid hormones readily crosses the cell membrane while most non-steroid cannot. As a result of this, receptors of steroid hormone can locate at the membrane or in the cytoplasm, while the receptor for most non-steroid hormone only locates at the plasma membrane. Since steroid hormones are derivatives of the lipid cholesterol, they are mostly insoluble in blood. In order for them to circulate in the bloodstream, they have to bind to certain carrier proteins to increase their solubility in blood. The carrier hormone complex is inactive, meaning that it cannot be accepted by any plasma receptors nor pass through the membrane. In order for the hormone to be active, it would have to disassociate from its carrier. Alternatively, one research published in Cell shows that the carrier hormone complex can also be endocytosed into the target cell and taken to the lysosome, where the carrier protein is broken down and the hormone is released. Once the hormone is disassociated from its carrier outside the cell membrane, it will either be recepted by plasma receptors immediately or pass through the phospholipid bilayer into the inside of the target cell. Once inside the cell, the hormone will be accepted by intracellular receptors located in the cytosol or the nucleus. Plasma receptors and intracellular receptors function very differently. The former is usually associated with rapid, non-genomic cellular responses involving signaling cascade, while the latter is responsible for slower genomic changes involving target gene transcription. Plasma receptors can be ligand-gated ion channels, G-protein coupled receptors, and receptor tyrosine kinases. Upon binding to their ligands in the cytoplasm, the intracellular receptors are disassociated from the chaperone protein that inactivates them and dimerize, or combine, with another ligand-bonded receptor. It should be noted that some receptors are located within the nucleus even before hormone binding and lack chaperone proteins. They are not dramatically different from those that possess chaperone proteins. The receptor-ligand complex then moves to the nucleus. This complex binds to the hormone response elementra on a specific hormone-regulated DNA, usually on the promoter part of a gene.